my name is Guadalupe Bolaños, and I'll be talking about adhesion junctions. So they're also referred as adherence junction, and they're just basically cadherin-based adhesive structures that mediate intercellular contacts between cells. There can be found just right below tight junctions, and their function is to initiate and stabilize cell-to-cell adhesion, regulate the actin cytoskeleton, maintain tissue architecture, and facilitate cell movement during tissue development and renewal, and they're involved in intracellular signaling and transcriptional regulation. So they're important in different stages of life. For example, in embryonic development, they promote the adhesion of identical cells and further organization and separation of tissues. And in adults, they maintain tissue homeostasis and control epithelial and endothelial paracellular permeability. And overall, they just promote cell-to-cell communication. So the structure involves core proteins like E-cadherins, P120 catenin, and alpha and beta catenin, vinculin and gamma catenin, as well as alpha catenin. Picture, you can see in adhesion junctions, an adhesion belt is formed, and it's held together by cadherins, and you can see that it is just right below the tight junctions. So these are just a bundle of actin filaments, and they're held together by, again, cadherin dimers. And the cadherin dimers, each is connected to the identical cadherin dimer from its adjacent cell. And the extracellular filament tails are held together by the anchor proteins, which are just basically the ones I have listed here, like the vinculin, the P120 catenin, alpha catenin, and beta catenin. So one study found that adhesion junctions are involved in the gastrulation process of a zebrafish, which is basically just the critical end stage of the development, where they just require their future body, like their anterior, posterior, and their dorsal and ventral axis, and their 3 germ cells. So basically, the E-cadherin, one of the core proteins found in adhesion junctions, is needed for the collective migration and the prechordal plate progenitor. And so basically, they just help modulate the correct migration of mesodermal precursors as a whole tissue, and they coordinate the convergence and extension movement of the YSL nuclei within the movement of the mesodermal precursors. There have been some studies that deal with adhesion dysregulation in cancer. And just because their adhesion junctions are responsible for a lot of major things in the cell, any irregularity of the system can cause transformation of cells and tumor invasions. So unregular expression of adhesion molecules causes dysregulated epithelial mesenchymal transition that has been linked to cancer progression and metastasis, where your primary tumors migrate to secondary sites in the body. So some of the findings I found was that decreasing the expression of P120 catenin results in abnormal regulation of cadherin turnover, which results in internal degradation of E-cadherin, which removes the capacity of the cell to suppress a tumor invasion. And also, the reduced expression of alpha and beta catenin has been directly associated with a poor clinical outcome and shortened patient survival for those who suffered from cancer. Overexpressing of a protein can also be linked to unfavorable prognosis, especially in lung cancer. And overall, the study stated that the deregulation of adherence junction genes can produce different outcomes depending on the type and stage of cancer in the body. So different expressions can cause different results. A study recently done actually in May of this year wanted to find the distribution and motility of E-cadherin, again a major protein in adhesion junctions, in punctate adherent junctions or PAJs of A431 cells. And they wanted to refine the super resolution description of cadherin's distribution in PAJs. So what they used was basically this fluorescent microscope, and they wanted to study them by staining them and in different solutions. So what they found was that A431 cells consisted of several cadherin clusters. The SMLM procedure revealed that each PAJ reached about 1 micrometer in length, and each cluster exhibited a dense and looser adhering regions. They also found that non-junctional cadherin is predominantly monomeric, and they are derived from the dendra-2 molecules. And adhesion junction plasticity is based on regulatable actin filament driven instability of the intrajunctional cadherin adhesive clusters. And so through fluorescent speckle microscopy and PAJs, they were able to see that the PAJs are indeed continuously and rapidly reorganizing cadherin molecules, so they're not just standing still. And a question I have is, are there currently any studies that are looking into the switch from E to N cadherin during the EMT and adherence junctions that can help target and diagnose different types of tumors? Remember that I mentioned that these irregularities in adhesion junctions can cause are linked to cancer's ability to continue to grow and infect the body more. So thank you for watching.